Welcome to another installment in our Biomechanics Closer Look Journal Club. This is our third speaker for this fall seminar series, and we're delighted to be welcoming new tenure track faculty member Arina Korneva. Just to give a little bit of a background about Arina, she originally studied in lymphatics at Georgia Tech during her undergrad. She went on to do her PhD at Yale University in cardiovascular mechanics. Then she transitioned a little bit into a postdoctoral position at Johns Hopkins, looking at nerve and optical mechanics, or mechanics of the optic nerve. And then she's now recently started her own faculty position at Virginia Tech in the Department of Biomedical Engineering and Mechanics, where she's going to be studying nerves and optical mechanics. So we're excited to host Arina today, and maybe I will yield the floor over to you, Arina.
Thank you. Well, thank you for coming on the call today and listening to the Journal Club, or if you're on YouTube, hello. So my first slide has the Virginia Tech colors because I'm presenting from Virginia Tech, but I'm going to switch to Johns Hopkins colors because the work was done at Johns Hopkins when I was a postdoc there in the Department of Ophthalmology with some affiliation with mechanical engineering. So I'll talk about my paper today. And I wanted to acknowledge all the co-authors, my postdoc mentors, and the funding from NIH, from the provost office to an undergraduate student, and from donors to the Glaucoma Center of Excellence. So thank you. So the overall goal in glaucoma, and my big picture goal for my lab, is to protect axons and repair them, the axons responsible for vision in glaucoma. And in glaucoma, the site of injury is the optic nerve head. And we've known for a long time that the injury occurs to axons around here. And we've known for a long time from clinical studies and from longitudinal clinical work and from studies on animal models that intraocular pressure is associated. And what intraocular pressure does, it expands the eye globe and applies load from the interior of the eye, and it also generates stress in the sclera. I apologize for the color. Let me try. It applies stress, generates stress in the sclera, circumferential stress. And of course, inside the mouse or inside the human, there's also some traction from the optic nerve, which extends all the way to the brain. And so somebody with a progressive level of vision loss would see something like this, where they wouldn't have a lot of peripheral vision and they would have a hard time focusing. And it affects 4 million people in North America and is a leading cause of reversible blindness, which is why it's very important to address that and find ways to protect axons. And so here's the way I kind of think about glaucoma in the big picture. So we have stress and strain with a predominant mechanical load being the intraocular pressure. And that will activate, we know it activates supporting cells. And there's some association to axons becoming damaged due to the intraocular pressure. And we know that with changes in gene expression and downstream signals, axons start dying back. It's called Wallerian degeneration. And we also know that with gene expression, there's some tissue and cell remodeling, and presumably that leads to changes to stress and strain and creates this vicious loop. So what we really don't know is how does pressure load the supporting cells and cause damage in the axons. And so in order to do that, we needed to perform a mechanical characterization of the tissue. So here's a little bit of background on this tissue here. So if you take a cross section in the Z direction, if you cut off the nerve, and if you're looking at this cross section here, I apologize for the drawing, but bring it here into this image. So these are the XY transverse plane. and you have a network of astrocytes. And these are enhanced images of the cytoskeletal network. And the cytoskeletal network is of astrocytes that are connected. So you could have one astrocytes that is spanning across the optic nerve head, or you could have some astrocytes that are hanging in different locations of the optic nerve head. And the axons aren't visible here, and that's because they travel in bundles, and the bundle is on the average a size of 2 microns, and the axons are going through the page here in the Z direction. And we have about 65, in the mouse, there are about 6,500 bundles of axons. And the material, so each dot here would be, could be an axon bundle that's going through the page with astrocytes providing structural and nutrient support for them. So now we get to the way we characterize the optic nerve head tissue, and that's by inflation testing. So we're interested in measuring the stress and the strain in the optic nerve head, ONH, and due to the intraocular pressure in intact eyes in 3 dimensions, and we're interested in it at the axon scale because axons are the ones that are being damaged. So we use 2-photon microscopy, and I enucleate the mouse eyes and place them on a plastic holder with the optic nerve head facing the objective. And the scan happens in the Z direction, and we acquire Z stacks from the 2-photon microscopy. And there's a needle that is inserted into the interior chamber of the eye, and the needle is connected to a pressure column, and the pressure, and we can regulate the pressure and calibrate the column to cause inflation or deflation of the eye. And so I wanted to briefly mention that while the rest of the focus of today's talk, because I only have 40 minutes, is on the optic nerve head here, it's going to appear, this stuff is going to appear black in the rest of the slides, and that's because we can't see it with one modality, but we can see it with second harmonic generation. So there really is, just remember that there's sclera surrounding the optic nerve head, and these are collagen fibers that are partly circumferentially aligned, as you can see here, around the optic nerve, and also some that are forming this basket weave orientation. And so while in the rest of the talk, I'm going to focus today on the fluorescent astrocytes, this will appear black, but just remember that they are surrounded by sclera in here. And so here's a maximum projection at 10 millimeters of mercury overlaid on a maximum projection, flipping with a maximum projection at 30 millimeters of mercury. So you can see there's deformation, but it is really hard to track by eye. And so instead of tracking it by eye, measuring the perimeter, we use digital volume correlation, which was established in the material science field and was a method in mechanics and has now really taken over a lot of the biomechanics measurements. So naturally, the original, or in quotation marks, original digital image correlation was done on speckled samples, but instead of speckling our samples, we use the endogenous signal from the fluorescence. And so I can give you a little example of what we see. So the algorithm that we use was developed by Christian Spronk's group, and the citation here, Barcoach by al in 2015, and it's available on GitHub. And the algorithm is a fast iterative Fourier transform algorithm for digital volume correlation. And essentially, it divides the image into a subset of a large window, and then it iterates and reduces the window size. And for example, one of the smallest window sizes that we get after the iteration is on the about 13 microns by 13 microns and by 2 microns. And so we get a very nice speckle pattern just from the fluorescence of the astrocytes. So in the end, we get a digital volume correlation after digital volume correlation, we get displacement about every 2 microns. So we get a nice dense grid by using the subset and having the next subset overlap it. So we get a very nice spatial resolution. And of course, after digital volume correlation in the 3-dimensional displacement field with grid spacing of approximately every 2 microns, we can calculate a green Lagrange strain tensor or any strain tensor you want with components EXX, EYY, or convert them to cylindrical coordinates ERR, E theta, etc. And so with a combination of the inflation testing, microscopy, and DVC, we can resolve the displacement to a grid spacing of 2 microns, which is about the average diameter of the axon bundle. And so in previous work, we asked what was the effect of remodeling here. So, we could inflate the tissue with we could raise the intraocular pressure, but then in glaucoma, we have some tissue remodeling. So what we found previously by using a mouse model of glaucoma, which induces an almost immediate rise in pressure for about 2 weeks, is in the previous work, we found that the pressure causes blockade of axonal transport, which is associated with axon damage almost immediately. And blockade of transport is associated with axon damage. So that's the biological process. And then from the mechanics studies, we found that at 3 days, the peripheral strain in the tissue, whereas it was previously compressive in controlled samples, after 3 days of exposure to intraocular pressure in vivo, elevated intraocular pressure in vivo, we found that the strain was no longer compressive at the edge and that the average EYY strain actually increased. So if this is the X direction, this is the Y direction, EYY strain actually increased after 3 days, and both EXX and EYY were no longer compressive at the edge. But at 6 weeks after exposure, where you do have a lot of tissue remodeling, we found that the peripheral strain was back to almost similar level, and the average strains were similar to the healthy specimens. So we had this characterization of the mechanics at different time points in glaucoma, and we needed to ask of how does that relate to the remodeling that happens. And there have been a lot of work characterizing the cell gene expression changes and the changes in the astrocytes in glaucoma. But we didn't know how to relate, and we wanted to relate it to the mechanics and figure out the crosstalk. So our first question was, does the cell changes or whatever remodeling is happening, does it affect the stress or strain? We found that it affected the strain in the previous work. So now it was time to unite that with some biological cell mechanisms. And of course, in the vast literature and this work in animal models going on for multiple decades, there are a lot of different mechanisms that were discovered and published, but we now needed to connect it to the mechanics. So after talking about it and looking at the literature and the work that was done by my PIs, we started to notice something interesting in the data. And the literature, including work in our lab, showed that at 3 days, the physical adhesions at the edge that are sort of breaking and the cells are retracting from the edge. So if you have this picture of an astrocyte cell with the sclera here, and the astrocytes have these end feet that are attached to the basement membrane. We don't really know what all of the molecules that compose these adhesions, which is why I'm not calling them, and the field doesn't call them focal adhesions, is because we actually don't know a lot about the molecular composition. But we and others have shown that within short-term exposure, that separates in glaucoma. And our lab found that at 6 weeks, the physical adhesions are reestablished. So my thinking was, okay, so there's something that is going on in the cells, and if we think about it, if the physical adhesions here are breaking, that's changing the boundary conditions that we have in the tissue. And so the tissue could have a more compliant strain response, and that could explain the lack of compression now at 3 days. And if they're reestablished at 6 weeks, that could explain, that compression appearing again at 6 weeks in the experiment that we have. So now it's time to test that. So the hypothesis was that the loss of physical adhesions in early glaucoma increases the strain response. And so how do we go on testing that? Well, um, the challenge was, we had to we wanted to do it in vivo, ex vivo. We didn't want to do it in vitro because as you isolate these cells in vitro, they take on a different morphology and have different biological signals. We wanted to disrupt the adhesions only. We didn't want to change the material in the sclera, so that was a challenge. We also wanted to do something that couldn't be toxic to cells, and we only wanted to target the astrocytes. We didn't, in the optic nerve head, we didn't want to target cells in the sclera, and we didn't want to target the large fibrillar the collagen fibers of the sclera. So after testing a lot of different, after examining different possibilities and having a background in biology, I figured out that we could use an enzyme called triple, and it's very similar to triple. Sorry, triple is very similar to trypsin, except for trypsin is derived from a pig a lot of the times when you buy it, whereas triple is a recombinant form, so it's not animal derived, so you can have a better, it's likely to be more pure in what you receive, and it's also likely to not have, so it's likely to be more pure and not have other proteases in what you purchase, and it's also was found to be more gentle on the cells. So it's commonly used in cell culture methods, especially with neurons or glia, because it's gentler and it's used to detach cells from the substrate. So to detach cells from the plates. So I thought that, I'm going to try to use it because I want to detach cells also from a basement membrane. So I wanted to detach the cells here in the schematic. So the hypothesis was that it would detach the cell's adhesions from the tissue edge and that this detachment will increase the strain response to intraocular pressure. So how do we go on testing this? Well, I wanted to use the same inflation test as we were doing before in order to compare our data. And then I devised a method so that we could add the solution with that enzyme triple or a control solution and incubate the eye for 60 minutes and then wash it out and then test the sample again. And it was important to do it on the same specimens so we could look at the strain response before and after treatment. So after what we did was we had these nice images of the astrocyte cells in green, and now I'm showing you the sclera. So this is a merged image, and what I did is I traced the area of the astrocytes at the before treatment and after treatment. So here and here. And what we found was, I don't know if you can see with me flipping the image, and this is the same specimen, but we have a small change in the area. So in 8 samples, we had a 10% contraction of the area, whereas specimens treated with a control solution had barely any change in area, it was 0.3%. Now, of course, that gets us to, is this a uniform contraction where the cells are stressed and now contracting all together, or is it really an effect on the physical adhesions at the tissue edge? So, looking at the data that we had, what I thought we could do was use our digital volume correlation technique and apply it to the specimens before and after treatment to measure to measure the effect of triple. So applied digital volume correlation. And so this is, I think, part where we, struggled with the terminology for writing the paper. And we didn't know, because we don't know if this is an active stress or if this is, how to define this, we said, let's use the calculations that we are using before. So the Green-Lagrange strain calculations, but now we want to really emphasize and make it clear that this isn't a strain due to applied loads. This is a a change due to the enzyme. So that's why we call it in the paper radial expansion, to be consistent with the direction, the positive in the Green-Lagrange strain. But I wanted to kind of show you here that in the strain maps, there was significant radial contraction in the periphery. So it was about negative 0.04, so about 4% contraction in the periphery, but it wasn't in the center. So if we had, in fact, a active contraction of cells, that would be, we hypothesized that if we had a active contraction of cells, the strain map would be more uniform, but we actually think that we had the contraction of or detachment of the adhesions at this edge here, which is why we didn't see the uniform pattern. We saw a lot of contraction at the edge and approximately 0 on average in the center. So that was really exciting. And also in the control samples, when we looked at the control samples in the paper, we saw the strain mostly on the order of the experimental noise. So we didn't see any contraction from the control solution. And also we prepared, these were measurements done during the experiment while the sample is in the holder and while it's being, treated and the cells are alive, et cetera. But then after the experiment was complete, we also fixed the tissue in paraformaldehyde and prepared them for transmission electron microscopy. And two specialists, Sarah Quillen and Dr. Harry Quigley, looked at the specimens under transmission electron microscopy. And in order to be more objective, defined 4 criteria for measuring the differences between control and the triple. And they were blind to the specimens, and they graded the images according to the criteria. And so after unmasking, we found that there were fewer adhesions to the basement membrane, and also, just lost my cursor, and also that there was discontinue. So if this is the control sample, the sclera is here, and this is the basement membrane here. And it's continuous and thick in the control specimens, but the specimens that were treated with triple, it's really thin and discontinuous. So this is the basement membrane here, and this is the sclera here. So this is the sclera. So that was really cool because this is the microstructural confirmation of what we are seeing in the macro images of the change in area and also the strain maps due to triple. So the one thing so now we confirmed the first part of hypothesis that triple detached the cell adhesions from the tissue edge. So here. So now it's time to test the second part of the hypothesis of that triple, by detaching the physical adhesions, would increase the strain response to intraocular pressure. So now we're comparing the strain response in this inflation test between these two blue dots to the strain response in the second inflation test. And wanted to point out that the strain, so we needed, because there's because there is some potentially plastic deformation here or some creep during this time, we wanted to, rule that out as an effect. So we looked as well at the samples that were treated with a buffer, so the same samples with the same loading history, and we compared the strain in this test and the strain in this test. So before treatment is in white, and after treatment with the buffer solution, control solution, is in blue. And we found that the difference was not statistically significant. And here, for example, was negative 0.008. So that's in the supplemental information. And in the EYY direction, the strain after minus the strain before was 0.0016. And all the numbers are in the supplemental information. So that was really exciting because that meant that we had a good control and we were getting a more or less reproducible loading of the eye. And so then we could use the data, the specimens treated with triple, and we saw that triple indeed increased the strain response to intraocular pressure, and that change in the strain was about an order of magnitude higher than the difference due to the control sample. So whereas this is within the measurement noise, this was significant, statistically significant, and the change in strain was about 0.0088 in the x direction, 0.01 in the y direction. So quite high. I think if you're starting really small strains, this is, several times more, several times higher strain after treatment with triple. And also, we are really interested in the study as it relates to glaucoma, because we found in the previous work that after 3 days of glaucoma, we had increased strain in the y direction. And actually looking at the magnitudes of strain, we found that our experiment reproduced the increase in the strain that was occurring potentially in vivo due to glaucoma. So this is data from a previous study where we had samples that were healthy compared that were inflation tested, and samples that were taken from eyes exposed to high intraocular pressure for 3 days. And in the first loading cycle, the strain difference was really on the same level as the strain difference that we had with triple. So that was really exciting. And we're not saying that triple enzyme is present in glaucoma. We know that it's quite unlikely. We are saying that changes in this mechanism that we were able to mimic in vitro, that mechanism of cell detachment could play a role in this increase in the strain. So that brings us back to this overall picture, is that we now have a much better idea of how the strain in this tissue due to intraocular pressure. We don't yet know how the axon's becoming damaged due to that, but we know some of the mechanisms that are causing some of the genes that are being changed, and some of the mechanisms of this in the cell scale of how they're being activated. And one of the mechanisms, as I said before, is that there's this loss of physical adhesions, at least in the 3-day glaucoma in the mouse, and that is reestablished after some time. And so this mechanism early on, now we know that it has changed the strain response, that it can change the strain response in glaucoma as well as in this in vitro setting. So in the conclusion, this work showed that digital volume correlation can be used to obtain quantitative and spatially precise measurements of changes in cell processes. So it can be applied to a number of studies. For us, it's really important because now we could use it in the future with other experimental conditions to measure the changes in the cell processes and how that changes. And we can use, our green Lagrange strain calculations or some other methods to quantify that. And the second part is that the reduction of the contact between the astrocytic lamina and the sclera, so the reduction of the adhesions, can contribute to the early increase in the mechanical response in the model of glaucoma. So our in vitro work has identified a potential mechanism that we need to explore further. And so that leads me to what I'm trying to do in my lab, in the neuromechanics lab, is that I've mentioned early in the first slide that the overall goal of the field is to protect and repair nerve cells and their axons from degeneration. But really, before we can protect and repair nerve cells and axons, we need to understand how axons are becoming damaged. So I've already mentioned that the optic nerve head, and what my talk today and the paper dealt with was the material that's in the optic nerve head here. So those were all the astrocyte filament network that was in the tissue here. And we got strain that were our spatial resolution was about 2 microns. So we presumably now have strain information of the material outside of the axons. But we really don't yet know the stress of that material and the material properties, and also how the axons themselves are being deformed due to that applied stress and strain. So that's the goal. The future goal of my lab is to figure out how axons in their native environment in bundles are being deformed due to that. And the goal is so if we can understand how they're being deformed and how that material is changing in glaucoma, we can figure out ways to protect them and make them more resilient to damage and make the material around them more beneficial to them. So thank you for listening, and I will take questions.
Great job. I was reflecting a little bit as you were speaking about, the past seminars that we've had, and I think this is the first, if not one of a few, I think it's the first ocular mechanics talks that we've had, which is cool. It's always nice to hear about a new tissue, new things applied to new tissues. So thank you for that. Anybody have any questions? Anyone from the audience?
It's funny you say that, Matt, because as I was advertising Arina's talk, I was thinking of people I could tag that do, optical mechanics or ocular mechanics. And I don't know whether there's just fewer people that do it or it's just not the community I've been part of, but I had a really hard time finding people. So that is just another reflection, I guess, confirmation of what you were saying, Matt. And Arina, extra thank you for being here and expanding our horizon.
Thank you.
I can start off. Maybe this is a super naive question, right? But you talk about axons, and the only context in which I know them is usually sort of brain mechanics.
Brain.
Are they similar? Are they the same? Is this sort of like a simplified model system of the brain? I'd just like to hear sort of my very naive perspective.
You asked me the $1 million question of what we don't know. So yes and no. So the reason I think that this optic nerve is really cool and why I want to learn about axons is because they can give us some insights into how axons start to degenerate in the optic nerve, but also in traumatic brain injury. And so the way that the axons start dying in glaucoma is there is some insult, and so they start to lose their transport So a lot of the molecules start accumulating here because they have transport blockade in this tissue. And transport blockade is also a mechanism that occurs in brain, in axons in the brain, and that is associated with subsequent death of axons. And the way that axons die is similar to the way they die in some instances of blast injury, spinal cord injury, is they die what's called dieback, Wallerian degeneration. So they start dying in the brain, in the nerve, and their cell bodies, which are in the retina, they are the last to die. So you can have visual impairment, but with the cells still being present here, which makes it nice because you can then, the field is really focused on trying to regenerate those axons. Because if you still have cells, that gives you therapeutic opportunity to regenerate the axons. And where I see myself fitting is, okay, some people are working on trying to regenerate the axons and adding stem cells to create new cells to grow new axons, but we don't know anything about the material in the nerve. So if that material is now different, how can we prevent further injury to axons? So my part of the, we're talking about biomechanics, and there's a lot of remodeling that occurs in the tissues of the body. So here we also have, in the optic nerve head, we have remodeling. In the brain, we have remodeling after TBI. Spinal cord, we have injury, we have remodeling.
Yeah, Johannes.
Yeah, thank you for this wonderful talk. I work on the brain, so I'm naturally interested in the similar things. And part of the lab is looking into the role of myelin in motivating brain stiffness. And I was wondering, how big is the role of myelin in glaucoma? Is it one of the key symptoms or pathological manifestations? Or is it just a side effect? And how do you have a way, or have you
Wait, hold on.
Go ahead.
I'm going to answer the myelin question. So actually, in human and in mouse, the area that gets injured is unmyelinated, and the myelin begins around here. So whereas later on, this material that surrounds the axon is myelin in addition to cells. In this region here, there is no myelin.
Okay, so myelin is not that important. That takes away my second question.
Okay.
Thanks for interrupting. No, it's good.
Yeah.
Does anyone else have questions for the speaker?
I would have another technical question if nobody else has a question.
Yeah.
I'm curious about the digital volume correlation. So the images that you've shown were primarily 2D images, right?
They're 3D images. It's just hard to show in the PowerPoint. Yeah.
Was there a particular reason you had to do a digital volume correlation rather than basically doing a 2D correlation? Like, do your features move in and out of your focus plane, or what was sort of where did the need for that arise?
They do move out, and you do have 3-dimensional deformation locally. On average, we didn't. So we started not knowing whether we'd have. We hypothesized we would have compression in the Z direction, and in the paper you can see in the data our strain in the Z direction was within measurement noise on average, but locally we do have some Z strain. So we do have 3-dimensional deformation, and that's why we use digital volume correlation.
Yeah.
Adrian?
Maybe Adrian?
Yeah, I have a quick question about composition of the tissue. So the outside you said is that we don't see is mostly collagen, and then these are mostly cells, and there's some adhesions in between them. But what is there besides cell? Is there some sort of also matrix that holds the cells together? And do we know anything about that composition?
Yeah, there's no fibrillar matrix in the mouse. inside between cells. There is fluid. So there is fluid transport, active and passive between cells, and nutrient transport between astrocytes and axons. There are glycosaminoglycans between the cell processes. That if that answers.
Yep, that answers the question. Yeah, thank you.
I might piggyback off of this question a little bit. So like the image that we're looking at here, you've labeled the fluorescent astrocytes. So could you clarify what actually is this labeling? What is this labeling in the cells?
Yeah, so it's an endogenous expression of green fluorescent protein, and it's driven by an astrocyte specific promoter. So
Okay.
we're using 2-photon imaging to acquire the signal. It is because the promoter is a cytoskeletal filament promoter. We think that's why the signal is localizing in the astrocyte processes, because we've seen I've looked at images from other astrocyte specific lines, and if the promoter is not cytoskeletal, it the fluorescent protein GFP doesn't localize with the cytoskeleton. Now we don't, it's not like attached to cytoskeleton or anything like that, and that's kind of the downside of fluorescence. So it's not specific to a cytoskeletal protein. It's just GFP in the cell.
Okay. And then I might follow up on the follow-up question. So along those lines, have you thought about, is it possible even to label the nuclei in here as well? So could you possibly see nuclear deformations inside, this area to see if that might have some effect on, the gene expression or any of these maybe downstream effects that you're seeing?
I don't. Can you rephrase your question?
Basically, number one, can you stain or label the nuclei? And then number two, can you use the DVC to track nuclear deformations?
I haven't tried. So I think that's more like a practical thing of theoretically you can stain nuclei. The thing is, nuclear stains are toxic to cells because they get in the nucleus. So the nice thing about this type of using a genetically modified mouse line is that the expression of the fluorescence, not a label, they're expressing GFP, and the cells are alive during the experiment. And also the second part of your question, if you stain them with a nuclear marker, you would have to change the microscopy quite a bit to use DVC. You would probably do like particle tracking, because you would have a couple of nuclei. So actually there aren't a lot of cells here. I could give you numbers after the talk. I would have to look up the numbers, but there are, I would say, like under 100 cells in this particular image. So in here. So you would theoretically you would get like nuclei here and there. And so instead of using a digital volume correlation, you would use a particle tracking algorithm. But if you wanted to see nuclear deformation, you would increase your imaging. You would have to increase your imaging resolution.
I see. Great. Any other questions? Yes, Mridang. Yeah, go ahead.
Hi, thank you for that great talk and congratulations on starting a
Thank
new
you.
tenure track position. So I'm gonna, I guess, like find one of the elephants in the room and be like, I see a lot of experimental mechanics here, and since my background is in computation, I was wondering if you have any intentions to do some sort of inverse finite elements on the optic nerve head to see whether the detachment leads to some change in material properties for the optic nerve head, whether the growth and remodeling actually changes tissue properties there. I was wondering what your thoughts were on that.
Yeah, do you want to do it? You can do it.
I think Manuel would kill me at that point.
As Mukang's advisor, I can give you a hard no on this one.
Yeah, so I think in my lab is transitioning to be an independent investigator. I'm transitioning into axon mechanics and nerve mechanics, but I know that my advisor's lab, Vicky Nguyen's lab, and Harry Quigley's lab, they are modeling this tissue in order to both estimate material properties, but also to figure out the strain contribution from the extracellular material, from axons, and from the different filaments inside astrocytes.
That sounds really cool. Thank you.
Can I ask one more question?
Can we do? Yeah, maybe after your question, if we could maybe stop recording, maybe people might be more comfortable with asking questions if we're at the end of the recording session.
Sure. Johannes, did you want to go ahead and ask? And then
We can, and I'm happy not to ask it if someone else wants to ask something without the recording. Otherwise, my question would have been, since you're starting a nerve mechanics lab, and especially if you're looking at the optics nerve, it is often used in clinical practice to evaluate the level of intracranial pressure, because it's really hard to measure that non-invasively. And if you have swelling, then the optic nerve is one of the easiest ways to assess or look a little bit inside of the brain. Do you have any ideas, or do you have any intention of looking into that, especially since you're interested in the optic nerve, of developing a model to deduce maybe the level of intracranial pressure based on short-term nerve degeneration or nerve changes, for example?
That's a great idea. So that I would love to talk more about that with you offline. I think I do. For the audience, I will say that yeah, so for others who don't know, we have cerebrospinal fluid here in between like this pink and blue area. So I didn't show on, I'm not showing in the schematic, but there is cerebrospinal fluid there, and that can be affected by intracranial pressure. There is a nice paper by Andrew Fiola in his postdoc with Rossi here that looked at intracranial pressure and optic nerve deformation in astronauts before and after flight. So that would be, I would direct the audience to that paper. Yeah, it's a really cool question in glaucoma of how the contribution of cerebrospinal fluid pressure and intracranial pressure on the optic nerve.
Thanks, that was really cool. And I'm happy to talk about this idea more.
Thanks. Can we stop the recording and then see if some folks